Okay, let's start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us, the 63rd seminar, and I call it a KU Leuven Day. And Happy New Year. And this is the first SynBYSS seminar in 2023. So I'm very excited. And I hope the holidays gave all of us time to reflect on 2022. There are lots of crazy things happened last year and spend with beloved ones, including family. Believe it or not, I haven't traveled for a month as of today and spending a lot of time with my family and friends in town. The last year was crazy with more than 160 days of seminar trips to give total 68 presentations and to meet people in person all over the world. And also, I hope you continue to move forward with your New Year resolution. And this is now already 12 days, and I hope you continue to do so. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Vitor Pinheiro. He is a professor at KU Leuven, Belgium. He is also quite a traveler. If I remember it correctly, and I confirm today, he is from originally Brazil and spent many years in Great Britain for degrees and as a lecturer. Now he's in Belgium. And I heard from him one day, one common thing is that the country names have letter B, like Brazil, Britain, and Belgium. So I'm curious what would be the next nation if he would move again. So additionally, I visited Belgium twice last year, including his current university, although he was in US traveling, of course.
Yeah,
unfortunately,
2 days last year I was in the US, you were here.
Yeah, exactly. So at that time, on my visit, so of course, I mean, we have something in common. And Vitor, I really thank you for your time and kicking off this year's seminar, and virtual podium is all yours. Thank you.
Thank you very much, Taisok, for the invitation and for inviting me even to come as one of the pioneers. So hopefully you should see my screen now.
Wonderful.
Brilliant. Yeah, so sort of for my 5 minutes, I wanted to go a little bit more about kind of how I got here. Because you often get this really polished story, and I thought I need to say a little bit about it, because he also partly explains how I got into direct evolution. So it's a real pleasure to also share the stage with Alon, who kind of came from one of the big guys in the field and now works in a former collaborators group. So it's quite fun. So in a way, kind of the goal of career now is really kind of reinvent biology one protein at a time, but you can also sort of describe it slightly differently. On the surface, as Taisok started, is really almost a traditional career. Did my undergrad in Cambridge, stayed there for PhD and postdoc, actually became a tenured postdoc in 2009, decided to start my own group, so moved to London and got tenured again in 2015. And through both kind of push and pull factors, ended up moving to Belgium. And now I'm tenured again, so third time lucky, hopefully. And recently also being promoted to Vice Dean for International Affairs. So people that want to exchange, both staff, students, kind of reach out. As a Brazilian, I should also point out that kind of there was a correlation in the number of coffees a day, but I'm trying now to contain, or maybe at least switch some of them for beer, since kind of now in Belgium, it makes sense. But in reality, kind of reality is a little bit more complicated. So of course, I would say my PhD may not have been the fastest way forward. I was effectively working bacterial pathogenesis, but for the postdoc decided to really go all out. High risk, high gain. Let's see what we can do with this. And certainly my time as a PI has been really much sort of learn by doing, which has advantages, but it has occasionally limitations. It obviously has not been a smooth path. So I kind of nearly walked away from science and academia a couple of times. And of course, the other things as well that do affect how things go. So certainly I'm kind of, I guess, generation 1 and generation 0. Generation 0 for moving abroad, generation 1 for being the first one in family that kind of is going above BA. And yeah, I have 2 wonderful sons, but it's hard work. So it comes tempered. But in that space, kind of I guess what merited me as pioneer was really the postdoctoral work. So I stepped into a project that was a really big question. So this was at Piet Irvine here in Belgium, Philippe Marlier in France, and Phil Holliger in the UK. They really tried to push this step forward. It's sort of, well, you have nucleic acids. We know they're obviously important for life. In fact, all life on Earth is DNA and RNA. But can you make something different? And it was a bit of a tour de force, and effectively over the last 10 years, we delivered 2 new ones, good ones. So hexitol nucleic acid there with that little extra carbon on the ring, and really a more challenging one, which is a threosyl phosphonate nucleic acid. So you have the shift from ribose to threose, but also this sort of phosphonate moiety. And the field has gone even further. We have sort of multiple modifications now available, and it continues to grow. But obviously, to get there, you have to engineer the proteins that do it. And certainly to shift something from DNA, and you can see here the kind of the standard classic B form of DNA, to something that might take this, like hexitol nucleic acid. And you can see here how well these the natural enzymes have evolved to fit around the nucleic acid. So it does become a challenge. And certainly at the time, and we're now talking so back to 2006, there were very few platforms for engineering this, and mutational data really only available for polymerases, and primarily in the context of applications, so primarily in the context of PCR. So it was a challenge. And of course, here's where I kind of got into direct evolution. So not kind of the flavor that Francis Arnold champions, but more the kind of the phage display side, more the kind of Jeff Smith and kind of the brought to the table. So really the idea that you can really sample a lot of the library and find what works. Of course, there are always pitfalls. As I went into sort of my group leader phase, the field was already starting the continuous direct evolution, but I feel that there's a little bit more to it. There's a lot of things that we can still exploit, and a lot of it has been focusing around here, because ultimately I do want to go to sort of demonstrate that an XNA world is possible, but the engineering challenge that every step is incredibly difficult. So certainly with the tools I have now, I cannot do it. I have to improve the tools, and we've been doing it. So really pushing primarily kind of the DNA assembly with some success in both polymerases and ligases. We're also looking at direct evolution to understand biology better, and that's coming with new plasmid origins, and a few more things in the pipeline that kind of hopefully in the next 2-3 years are going to see the light of day. In addition, kind of I was there to help start as sort of the SynBYSS community in the UK. I actually ran the SB UK conference in 15 and 16, but now in Belgium, we're going to do it again. So we started with a kind of the possible consortia. So Marianne de Meij, Evelien Peter, Sarah Leber, Kristen Hens, Kevin Verstrepen, Rob Laving, and myself. We are really representing 4 different universities here in Flanders, so the Flemish side of Belgium. And that will be the core kind of to take on to the next step. And there's still, there's incredibly competent, sort of excellent microbiologists in Wallonia, which is the other side of Belgium. And we've got to tap into that to something interesting. Now, our project, because we have to really build the community from the ground up, has been to look at kind of model microorganisms and really try something new. So try the idea of these universal genetic circuits, so that we can then expand into non-model organisms. And setting all of this up into a really ethics by design for SynBio. So capitalizing on the strengths of Belgian science to build really a SynBio community in Belgium. And with that, kind of, I also need to thank everyone who supported the idea, supported sometimes quite critically, but did it kind of help it get this far. And of course, everyone that has been through the lab, from master students to PhD students and postdocs, that have made everything here possible. So with that, thank you very much. And I guess back to you, Tasok.
Oh, it's amazing, amazing. You know what? I also found the very interesting coincidence. You showed 4 cities, and that city I actually went to when I visited twice in Belgium last year. So that's very also fascinating. Also, one thing you mentioned that resonated me a lot. You said, some struggle in the early career in your life. And I also talk to young people, and one thing I always tell them is basically fail miserably when you are young. And out of that, out of that failure, we learn a lot. I actually failed when I was young, and then I learned a lot from that experience. And then when you become older, I mean, it's so difficult to overcome out of failure. But, when you are young, I mean, we can start again. So that is fascinating thing. And then, also as an international student, there are multiple more challenges in general. So, but you overcome and then become one of the pioneers. So that resonate with me a lot. So I thank you for sharing that story. That's brilliant. Okay. So now the main speaker of today with a longer introduction. Dr. Alon Wellner completed his PhD with Professor Dan Topfick, where he studied basic aspects of protein evolution. Another, same, topic, evolution again, using directed evolution and structural biology. As you may know, Professor Dan Topfick was a professor at Weizmann Institute of Science and has tremendously contributed to the scientific community. Sadly, he passed away in 2021, of course, big loss to us. And after 5 years of leadership position in Israeli biotech startups, Alon basically joined Changlu's lab for synthetic evolution at UC Irvine. During the past 4 years, Alon has developed a system that allows rapid discovery and engineering of antibodies by combining orthogonal replication with yeast surface display. This is a unique career with a combination of academic training and working experience in startup companies for many years, which makes him a truly rising star. Alon, thank you so much for your time today, and please take it away, and everything now yours.
Thank you so much, Tatsuk, for the kind introduction and also for organizing this and inviting me today. I'm super excited to be here today and for this opportunity. I'll just quickly share my screen. start the slideshow. You can see it?
Yes, perfect. Thank you.
Okay, so yeah, again, thank you very much, and I'm very excited to be to tell you today about my work that I did during my postdoc. Some pictures from my traveling here in California with my family. So yeah, I've been a postdoc here for the last 4.5 years almost, and I've been developing the system for rapid generation of antibodies using autonomous hypermutation in yeast. So it's really hard to overstate the importance of antibodies in biomedicine and in life science due to their exquisite capabilities to specifically and strongly bind to their targets. Antibodies have been considered the magic bullet in the treatments of various diseases, especially such as infectious disease, notably SARS-CoV-2, Ebola, anthrax, and others. Transplant rejection, one of the earliest indications, actually the first monoclonal antibody drug that was approved in 1986 was for transplant rejection. Obviously, lots of cancers with immune check inhibitors, and also discovering antibodies to arm T cells for immunotherapy. And as we all know, autoimmune disorders, the Humira, the best-selling drug of all time, is an antibody. So that brought lots of help to many patients around the world. But still, the vast majority of antibodies today are still discovered using traditional methods. And this is mainly animal immunization. In this graph here, you can see that it's been 35 years since the approval of the first antibody drug, and the rate of approval of these types of drugs has been increasing exponentially. But still, the vast majority of them are still discovered using animals. There is more usage of transgenic animals, but only 10%, only 10 out of the 100 drugs by the FDA today have been discovered using synthetic libraries, using all kinds of in vitro systems like phage or yeast surface display. And this is really not surprising, given the fact that when you introduce a foreign antigen into an animal, you exploit this very powerful response of the germinal center, where B cells are first selected for clonal recognition, for antigen recognition, and then they undergo, again, the very powerful response of somatic hypermutation and affinity maturation, where they increase their specificity and their potency against their targets. So it's really hard to compete with an animal's immune system. However, there are many issues associated with animal-derived antibodies. It's a very slow and expensive process, and that makes it largely inaccessible to people like us in academia. If you want to raise an antibody against a target that has to do with some aspect of your research, there's a really high bar. And also for therapeutics, there's a really high bar to entering the market. It's also a very uncontrolled selection process. You can't really control which epitope in your antigen you are targeting. And this is something that you can much more easily do with in vitro systems. It's inaccessible to self-antigens due to immunodominance and self-recognition, and also to complex antigens such as GPCRs or other membrane proteins. And this is especially true for when you want to raise an antibody against a specific conformation of a GPCR, that you would need to lock that conformation by complexing the GPCR with its ligand. So that complex would probably dissociate when you inject it into an animal. There's also many ethical and reproducibility issues associated with animal immunization. So synthetic or in vitro systems for antibody discovery and evolution do exist. And those are mainly phage and yeast surface display. And these have been the working horses in the field for the past 20 years. Both of these systems have their own advantages and disadvantages. So for instance, in phage display, you can introduce relatively large libraries due to the very efficient process of E. coli transformation. In yeast surface display, you can differentiate between the titer of an antibody on the cell surface and between its affinity by using flow cytometry. But eventually, the problem with both of these systems is that you're ultimately limited by the initial complexity of the library that you're introducing into the system. So these libraries are static and they don't continue to evolve. And this very much limits your sequence space exploration. Obviously, you can also use these systems for directed evolution. You can find a hit and then you can diversify it using some kind of error-prone PCR or gene shuffling or whatnot. reintroduce it as a library into phage or yeast, and then run another panning or phage selection. And then you would have to reiterate this process again and again, typically running several rounds. And each round would take you between several weeks and several months. And it's really hard to parallelize this. So an ideal system in our minds for antibody discovering and engineering would be based on an in vitro display technique such as phage or yeast, but the antibody encoding gene would be continuously evolving, exactly like what's happening inside B cells. So our idea in the beginning of this project was to generate some kind of a synthetic B cell, or something that would function as a synthetic B cell using yeast. And for this purpose, we have designed and developed the following system that we call AHEAD. It stands for Autonomous Hypermutation Yeast Surface Display. And in AHEAD, AHEAD is based on the yeast Saccharomyces cerevisiae. It harbors a very special plasmid that is called P1. P1 is a linear plasmid that sits only in the cytoplasm of yeast. It's a linear plasmid that's kept by these two terminal proteins, and it's replicated by its own orthogonal DNA polymerase. And the reason there is complete orthogonality between the replication of this plasmid in the cytoplasm and the rest of the DNA inside the nucleus is that this special DNA polymerase has to first interact with the terminal protein. So there is no replication by this polymerase and nuclear DNA. And this polymerase has been engineered to have a mutation rate which is 100,000-fold higher than the mutation rate inside the nucleus, but it does not affect the mutation rate inside the nucleus. So it replicates P1, and you clone your antibody gene fused to the AGA2 cell wall anchoring subunit that allows the antibody gene to be displayed on the cell surface. And this defines a very streamlined process in which all you have to do to build a library is simply to clone this antibody onto P1 and grow a culture of these yeast. And when you grow the culture of the yeast, the antibody automatically gets diversified. And so just by growing yeast for a couple of days, you get a library. And once you get that library, you can mix it up with some fluorescently labeled antigen. And then you can use phage selection to enrich for mutants of this antibody that have increased affinity towards the antigen. And then the cool thing is that once the cells recover from the phage selection, they automatically rediversify your antibody. So all you need to do is grow cells and phage them, grow cells and phage them. And this whole process is very short. It can be done in about 3 days with very, very little effort associated with it. So you just grow the cells, phage typically takes like 30 minutes. So you can imagine how it's very easy to parallelize this experiment and to perhaps evolve many different antibodies against the same antigen or go after various antigens at the same time. So one of our first proof of concepts was in the beginning of the pandemic. So it was very obvious for us, straightforward to focus on the SARS-CoV-2 trimeric spike protein as an antigen, specifically the receptor binding domain of it, the RBD. That's the part that latches onto our ACE2 receptors, and that's where we want antibodies to neutralize it, to prevent or to treat infection. And as a antibody fragment modality, we chose to focus on nanobodies. So nanobodies are these small variable chain only fragments from camelid IgGs. So these are 15 kilodalton globular proteins that have similar binding properties as conventional antibodies. And they're very small, so that makes them very easy to engineer and to express in bacterial system and in yeast. So this is how a typical affinity maturation campaign using our system looks like. In this example, I'm showing a nanobody that we call RBD1 that binds RBD. And the RBD1 initially had a very low affinity of about 1.4 micromolar. And indeed, when we stained this nanobody, the yeast displaying this nanobody with a lower concentration of RBD, we couldn't detect any binding, but nonetheless, we could sort some mutants that were in this gate. And in the next round, after 3 days, we got this new population that signifies a mutant with an enhanced affinity towards RBD. And then again, we sorted them, grew them, and then you can see that in every FACS round, you typically would get a new mutant with an increased affinity. So you just accumulate mutations through these FACS rounds. This whole process took about 2 or 3 weeks with very, very little work associated. All I needed to do is grow the cells, fax them, grow the cells, fax them, and that typically results in the accumulation of 4 to 5 mutations and increase the affinity by anywhere between 1 and 3 orders of magnitude. So since it was so easy, we could, in parallel, affinity mature 8 different nanobodies. All of them increased their affinity towards RBD. And the scheme here of a 2-dimensional scheme of a nanobody, you can see that these are the mutations that we've accumulated as expected. Many of them were found in the complementary determining regions of the nanobody, but also about half of them were in framework regions of the nanobody. So all our nanobodies increased their affinity towards RBD. I'm just showing here some notable examples. So this is the nanobody I showed before, RBD1. It increased its affinity by almost 2 orders of magnitude. This was our best performer, RBD10. It had an initial affinity of about 0.5 micromolar, and it became a sub-nanomolar binder. And also, all our nanobodies were also potent neutralizers of SARS-CoV-2, shown here by this pseudovirus neutralization assays. These neutralization potencies are among the highest reported in the literature for nanobodies. And also, all these potencies increased from the parent nanobodies towards the evolved ones. But we noticed here some interesting trend. So for instance, RBD10I14, our evolved versions of RBD10 that had a sub-nanomolar affinity, was actually a pretty modest neutralizer. While RBD11I12, which was actually quite a weak binder, was our most potent neutralizer. And that led us to think that there might be some functional diversity between these nanobodies. Maybe they're binding different epitopes, and this is why they're neutralizing it differently. So to test this, we've devised this ACE2 competition assay that was based on biolayer interferometry. So this assay goes like this. The first step is to bind our biotinylated RBD to a streptavidin-coated BLI surface until we reach saturation. Once we reach the saturation, we flow in the nanobody again until we reach saturation. So it looks like this. And then we flow in ACE2. If ACE2 and the nanobody do not compete over the same epitope, you will get an increase of the BLI signal. While if there is a complete competition between the nanobody and ACE2, you will not get any further BLI signal. And these two here are just known controls that we took from the literature of antibodies that either compete or do not compete with ACE2. And this is also validated by cryo-EM structures. And indeed, we saw that nanobody RBD1I13, which was a relatively modest binder or a very potent neutralizer, completely competed with ACE2, while RBD10I14, which was our most strongest or sub-nanomolar binder and very modest neutralizer, did not compete at all with ACE2, and RBD11I12, which was again a very weak binder, but a modest, but a good neutralizer, somewhat competed with ACE2. Again, this strengthens our conviction that these nanobodies probably bind RBD at different epitopes. The ideal thing to do at that point would be to co-crystallize these nanobodies with RBD. We're not a crystallography lab, so instead of that, we decided to use a synthetic approach using a deep mutational scanning library. This is a library that we got from Jesse Bloom's lab at the Fred Hutch in Seattle. So in this library, they built a deep mutational scanning library of RBD in which every position in RBD is mutated to all of the other 19 amino acids. And using this library, you can add an antibody on the library and select for the escape fraction. So you're actually selecting for the RBD mutants that cause the RBD to escape from antibody binding. And you can compare these two NGS runs and get these kind of logos that signify what is the escape fraction. And this can give you a rough estimate of where the antibody binds, because if a position in RBD mutates and does not allow antibody to bind it anymore, it's probably where the epitope sits. And indeed, this was in quite a good agreement with our previous findings. So RBD 1I13, that modest binder but very good neutralizer, so its binding epitope seemed to be in the vicinity of the interface between RBD and ACE2, while RBD 10I14, which was a very strong binder, weak neutralizer, it seems to have like a larger epitope, but this epitope is a bit more distal to the interface between RBD and ACE2, and again, the same story for RBD 11I12. And we could also see that it covers different mutational landscapes on the RBD with known mutations that cause escape from antibodies, and these mutations, especially position 484, L452, 490 are known mutations in variants of concern that later arose that were escaping immunity from vaccines and from prior infection. So by our ability to parallelize affinity maturation to raise different antibodies in parallel, we could target different epitopes on the RBD, and you can imagine how maybe in the future this technology can be used to generate quickly generate cocktails that can prevent escape from immunity. So just to summarize this part, our head system can be used to rapid affinity maturation of surface displayed antibodies. I haven't mentioned this, but I've shown nanobodies, but we can also do the same thing for SCFPs and also for Fabs. And since it relies on simple cycles of culturing and selection, these experiments can be highly parallelizable and accessible. And these parallel affinity maturation experiments increase the potential of discovering diverse set of antibodies. So up until now, I've showed you what happens when you clone a single antibody fragment onto the system and affinity matured, but you have to get that antibody from somewhere. These antibodies, we got them by screening a synthetic library of nanobodies, but other antibodies that we've used or other used with the system came from animal immunization or phage display. But our ultimate goal with the system is to generate an autonomously evolving naive library. This could be done for other fragments, not only nanobodies. Currently, I'm focusing on nanobodies, but the idea of having such a library is that once you clone a naive repertoire of antibodies into the system, it self-expands. So you can potentially expand this library and get like an ultimate, like an unlimited diversity. The other thing is that this library can sort of function like a synthetic immune system because it has these two components of clonal selection and then affinity maturation, and it can be a very useful tool for the community. We can ship this out to anybody that wants it, and it could, imagine just having a tube like this, having your antigen in several rounds, you could just get a very potent antibody. It can be a very useful thing for research. The other major advancement that we recently had in the lab was that, and this was done by a very talented grad student, Gordon Ricks, and it was assisted by Rory Williams, a new postdoc in the lab. They both engineered new polymerases now that actually have a 15-fold higher mutational rate over what we previously had. So our previous polymerase called 633 had an elevation mutation rate of about 100,000-fold. Now we have about 1.5 million fold higher mutation rates. And this really is a game changer for us. The other thing is that previous polymerase was very, very limited in terms of the mutational scope. It mostly made transition mutations, and now we can also get about 20% transversion mutations. So the scope of mutations that we also get now is much more increased. So almost any amino acid can turn into many other amino acids. So this was a very big game changer. I've tested what happens with these new polymerases, and for doing this, I did this experiment where I took RBD10, the nanobody that I showed before, and I did this very meticulous comparison between them, where only the top 15% displaying cells were selected, and then something like 0.1 top display binders from them were selected. And then I ran this for 3 rounds. The result of that was that these new polymerases functioned much better. And you can see that the binding affinity that these populations gained after 3 rounds were much higher than the previous polymerase. And also, they accumulated many more mutations. The mutations that are signified here in orange are result of transversions. So armed with these new polymerases, I've decided now to integrate a new library into the system. As a library, I chose to use this synthetic nanobody library that was generated by our collaborators in Harvard Medical School, Andrew Cruz group. This is a very clever design in which they used a single framework modality, and they diversified only the CDRs based on multiple sequence alignments of nanobodies that are found in the PDB. And the idea behind that is that nanobodies that have crystal structures are generally biochemically well behaved and have good therapeutic properties. This library has already been shown to generate very potent antibodies against hundreds of different targets. So I cloned this library into our system with the new polymerases that have a higher mutation rate. And I've ran several affinity maturation campaigns against some pilot targets. And this is just to demonstrate, this is actually kind of like really recent data that I got. So just to show after like only 5 rounds, you can get pretty potent binding against a very low concentration of RBD. In this case, I decided to use the RBD that's 17 mutations away from the wild type one, Omicron BA5 version, and another pilot antigen, CD3 epsilon. That's a very appropriate target for antibodies. IL-17, another cytokine that is involved in autoimmune disorders. So you could just take this library, get your antigen, and very quickly, within a couple of weeks, you can get like a pretty strong binder. I just ran some EC50s last week on yeast, and these potencies that we get here are binding affinities are probably underestimations of what we can actually get when we're going to measure these with SPRs. And these are the mutations that we've accumulated from the parent clones that we isolated from the library. So this is just to demonstrate that you can take this library, take an antigen, very quickly evolve an antibody with many mutations to bind pretty strongly to that antigen. So what's next? So I'm about to wrap up my postdoc and thinking about opening my own lab. And I want to use the system to generate not only binders, but actually functional antibodies and an appropriate class of targets. I think for the system are GPCRs. So GPCRs are a very important class of targets. They're associated with actually every aspect of human physiology. There's an increasing trend recently of GPCR targeting antibodies. There are more of them targeting, getting into like clinical studies and even approved for clinical usage. The idea behind that is they can have much better pharmacological properties over small molecules, but they also can be much less toxic. GPCRs tend to be very conserved between classes in their binding pockets, but antibodies can have a much larger binding epitope. They have much more real estate that they can cover, so that can generate more selectivity towards targets. And what I ultimately want to build is a system that can actually directly select for functionality, not only for binding, and that also prevents the need to isolate the GPCR, which can be very hard to do sometimes. So the system that I'm planning to build would be based on microfluidics, in which a secretor yeast cell, so I can turn these yeast surface display into cells that just secrete the antibodies instead of displaying them, would be co-encapsulated with the mammalian reporter cell that can report on GPCR activation. There's like different ways to do that. This is just like one example, but this can be done also with a regular Tango assay. And one of the first targets that I'm thinking of targeting would be the mu OPRID receptors. So there is pretty convincing evidence that their analgesia could be achieved by triggering mu OPRID receptors in the periphery, and antibodies have this very built-in property that they cannot traverse the brain-blood barrier, and this is why they wouldn't have toxic effect on the central nervous system. But this can be actually translated to any type of class of GPCR, and so with this, I would like to finish and thank you very much for listening, and to thank all my lab members, and especially Chang for being a great mentor during my postdoc, and all our collaborators from Harvard Medical School, especially Andrew Cruz. And yes, thank you very much. I would be very happy to answer any questions.
Amazing talk. Oh my goodness. I mean, this is a lot of challenging work you already accomplished, and I really learn a lot actually. And Vitor, do you have any question while we're waiting? Any question from audience?
I have many. But Alon, maybe one question I was quite curious about is what kind of insertion and deletion rates do you get with these polymerases?
I haven't seen any.
Okay.
We don't.
Yeah, because I guess you need 3 for it to come up again.
Yeah, we're we're just diversifying the length of the CDR3. So when we build these libraries, we just have like different lengths. But it's based on orthogonal replication, so it's just mutations. You don't get in those.
Vitor, any other question or comment?
Well, I'm quite curious because I mean the system you're proposing here at the end, I mean that's quite intense to kind of co-compartmentalize in a way that still grows, secretion still works. I mean, is it feasible at first just to kind of express those antibodies inside the yeast and see if you can modulate a yeast GPCR? Because then you can tap into a circuit where if it gets activated, it changes color. So you can kind of go into the facts, but sorting just the population that is functional.
Yes, definitely. I mean, you can express GPCRs in yeast. I think, there's like examples, probably like half of human GPCRs can be expressed on yeast functionally, and you can read out the signal. So that can be like combined together. The challenge here would be that the GPCR would be expressed on the membrane, and if antibody is being secreted, then it has to go through the cell wall. So I'm not sure how the cell wall of the yeast would be permeable to antibodies, but perhaps there could be some kind of a trick where the antibody is being secreted from the membrane, but stays in the interface between the membrane and the cell wall, and trigger the GPCR, or maybe even somehow displayed in the inner part of the cell wall, or just from the membrane to trigger the GPCR.
Yeah,
and then that kind of like makes the whole microfluidics redundant, right? You could just like phage yeast.
Because then if actually growing and putting the selection on survivability, if you have a kind of some kind of life death circuit running.
And that's really easy to like to connect between a GPCR activation on yeast and just a GFP signal or survival, or maybe survival could be better in that case. You don't need phage, and your throughput actually increases.
Yeah, pretty cool.
You could continue. If you don't have, I could ask some question, but you could continue, Vitor.
No, yeah, I think because the other questions that become technical, I'm not sure if the attendees are keen on listening to that, because it's all about, the continuous evolution. In theory, you have this ability to sample the entire landscape. Realistically, you're dependent on that polymerase and the error profile of that polymerase. So in a way, have you ever tried to map out the neutral mutations you're getting after kind of a certain time of growth, or kind of have you tried? So effectively, I think the question I'm sort of coalescing to is this just error-prone PCR on steroids?
In a sense, yeah, it is an error-prone PCR on steroids. Previously, it was very biased toward these transitions, and now we're like super excited about these new polymerases because we're starting to see mutations that we've never seen before. Like alanines can turn into glutamates. Like the entire codon table is opening up for us. But yeah, it's an error-prone PCR without you having to do it. Right? I mean, you just grow cells and it happens inside the cell. So that's super exciting. Yeah, and our another goal that we have, but that's a struggle. It's been a struggle for the past few years, but we're actually having some ground covered on that front, is decoupling the selection from phage. Because now it's not really continuous in the sense that, when you put an enzyme that has some kind of function that's linked to survivability of the yeast, you can just grow the cells and just increase the selection pressure, and then the orthogonal replication and the selection are coupled together. In our case, it's kind of like semi-continuous because you do have to stop growth and conduct selection using phage. So you're not selecting all the time. Imagine if you had like a system where the cells are always under selection for higher affinity of the antibodies that they display. That would be ideal.
And in that sense, can you coat the inside of the growing tube with your protein? So just rely on retention in the fermenter.
Yeah, that's a good idea. Yeah.
I don't know how well it would work, but
yeah, that could be a cool idea. It's hard to think how would you like increase the selection pressure, but I guess there's always going to be a selection.
Yeah, because I guess.
Well, maybe some cells will just start falling off right from the surface, and other cells with higher affinity will compete with them. Yeah.
So effectively, because that's how you work with aptamers. You can only punch into the really high affinity when you start to get competitional off rates. So you would have that drive into that space. Yeah.
So the other idea that we're going after is to try to link the affinity to growth by hijacking the yeast mating response. So, yeast, when the A and alpha yeast, they come together to form a diploid cell, they first have to agglutinate. And it's a known fact that you can replace these agglutinin proteins with synthetic agglutins, and that the propensity for two opposite mating types to form a diploid depends on the affinity of that pair. Eric Clavin, in University of Washington had a really nice demonstration. They started a company that's very successful called A Alpha based on that, where they can just like make libraries of protein-protein interactors and very easily just like measure the affinity between interaction partners. So by replacing these agglutinins with like an antibody and an antigen pair, and we actually we're able to do that. So we showed that we can change them into an antibody and antigen, and they would fuse, and that fusion propensity is correlated to the affinity between the pair. The challenge here that we're facing is how to turn this into a signal that causes the cell to propagate faster.
Yeah, because I guess you need the antibody, but you need somehow the signaling going in as well.
Yeah, yeah. So yeah, we initially thought that it could be done using pheromone signaling, because the cells first come together and then they signal with the A and alpha factor to fuse together. But you don't want them to fully fuse, that because that's a dead end. And the problem is that the pheromones, they kind of diffuse into culture, so it's hard to get like a so maybe with microfluidics or some other trick.
Yeah. Ooh.
Work in progress.
Yeah. Yeah, that's a tough one. Oh, that's cool.
So any other, because I seeing no question from audience, so you could continue or I can ask question.
No, I'll say have a turn.
Okay, sure. So, I used to do protein engineering and love directed evolution. But I have one trauma that is basically I was human machine picking 40,000 colony by myself, picking, and then doing color based assay myself. So I was the liquid handler when I was a grade student. And after that, I decided not to do any protein engineering, but recently I studied protein engineering because of all the interesting tools, including AlphaFold and so on. And naturally, your case, it completely makes sense to use directed evolution because you have the very nice screening system. But now I will also see some paper, they use machine learning, also this kind of computational algorithm to use protein engineering and even antibody engineering. So I just wanted to hear your perspective on this kind of complementary kind of approach now happening in the field.
Yeah, I think it doesn't necessarily compete. It actually is complementary.
Yeah, complementary.
Yeah, because for machine learning, what you mostly need is good training sets, right? And I think that using the system, we can generate large data sets for training algorithms. And now we have this ability to put large libraries into the system. It was actually quite a big challenge because just like introducing libraries into yeast is already a big challenge, like large libraries. And in our case, the efficiency is about 100-fold lower. Because we have to cloning onto this P1. I mean, I just said that we're doing that, but I haven't explained how. It actually depends on some kind of like a weird recombination events that happens inside the cytoplasm. So we transform DNA. That DNA has to recombine onto like this landing pad P1 in the cytoplasm and that's a very, very inefficient process. But we've optimized that and we brought it to like a reasonable level. So we are now able to like introduce relatively medium-sized libraries into the system. So I could envision like a good combination between machine learning where we just generate large amounts of data, can go after different targets. Because we can parallelize, we can also do negative selections against like polyreactivity agents, for instance. We could we can gauge lots of different parameters of not only affinity but also polyreactivity, stability, stuff like that. Generate large data sets and feed them into machine learning algorithms, and maybe design new libraries based on that and have like a cycle.
Interesting.
Yeah, but can I push the caveat in there? Because I
Sure.
think what Alon mentioned is the key thing, is having the good data set for training.
Right.
And if the protein is not highly correlated, point mutants, double mutants, that will deliver the kind of data set you need. But if you have highly correlated positions, which sometimes can happen in an enzyme, and here I'm really thinking about polymerases, that data is not good enough for training. So
Right.
either you have to have a system where you can continuously feed new data, or we need to really look into what or how to go about designing a good training set.
Right. That is exactly what similar to what I envision. So I'm not doing those kind of antibody work, but what I envision is basically the current system I believe you are using is basically selection, but you are not generating real data. But what you could do in the future when you start your lab or Chang Lu's lab could be that takes more time, but you rather than just only selecting things, but you also do some sequencing coupled with the fluorescence data. In that way, you may have data, sequence, and whatever activity sort of kind of data set, and that generate a lot of data. And then after that, you may have machine trained with that data, and then at the end of the day, you may have some predictability in the end, and that is something I kind of thinking about. In that way, you not only just keep screening, you could have one principle, some correlation, and then the machine will be useful for other applications as well. So that's kind of something I'm thinking. But yeah.
Yeah, sorry, Alon. If I answer this one first, I totally agree. We've, as far and as wide as we have managed to do, we've switched to next generation sequencing to be able to access that kind of data. And in some cases, it has paid off. So we were trying to evolve a mesophilic HNA polymerase, but in fact, looking at the data and then further characterizing the enzyme, what we saw is that we can also use directed evolution to understand biology. But it hinges all on the data analysis.
Right. So I see Maya raise your hand. Maya, do you have any question or comment?
Yeah, I mean, this ties into what you're saying a lot, but I am curious how much within the sort of field of directed evolution, how much is this expected to be the sort of directed evolution itself as an end in terms of, when you want a protein every time you do directed evolution, and how much of it is expected to be a means to an end? And the assumption is that we get enough data that we can do one-shot design.
So that's question to whom?
To
Alon?
I guess, but I mean, anyone can have an opinion on this. So whoever wants to have an opinion.
So Alon, why don't you start?
I mean, ideally, if eventually we could just like have these very good algorithms that, just like design antibodies without our need to do experiments, that would be great, right? I mean, I'm not sure that, I think it's always going to be a combination between the two. Maybe eventually down the road, we'll just have like these perfect algorithms that will design anything like antibodies and enzymes, but it's hard for me to predict at this point. Vitor, what do you think?
No, I'm in the same boat. I mean, I think eventually we will converge into a good enough system that you don't have to do the experiment. But for now, experiment is unavoidable. And I think I would go even one step backwards. I mean, I don't think we have the right tools to analyze how good a selection platform is, how to compare different selection platforms for a single target or even between different targets. And yeah, I mean, if you have a continuous system, so a system that's continuously sampling the sequencing space, you can get away with things. But if you're still on a turn-based or a screen-based, that's there's a big hole there still.
I feel the same way. I believe directed evolution and computation-based engineering will continue in a complementary way, because biology messy and then too complex. And then you could have predictable engineering for one protein, but same approach could be applicable to another protein, because we need to run again. So at the end of the day, you know, we may reduce the number of screening, but at the end of the day, I guess still combination of predictable algorithm-based design and then screening and some directed evolution. That is actually, you know, the entire field for many decades already done in terms of enzyme engineering. They did computational whatever prediction and then design something, and then after that improve the enzyme activity by directed evolution. That's even 20 years ago, you know, people already done that, and then now still people kind of doing that. So that's my feeling, but who knows? I mean, one day perfect algorithm, I don't know, but probably not in my lifetime, probably next generation.
Yeah, definitely to reach that goal, we need like really good training sets. And I think, you know, good directed evolution platforms. And, you know, I'm super excited about our platform just because we can just like, you know, take the same thing and go after like lots of different things in parallel. It's really hard to run a directed evolution experiment because you have to manage these libraries. You have to like every round, it takes a lot of work. You know, just like we could just like do these experiments in parallel now. Take the same antibody and just like have different selection pressures posed on it, get different outcomes.
Sure.
Yeah.
Okay, good. Now I see no other question, and I see now that is the 11 o'clock basically pass. I mean, so why don't we wrap up and then we chat more, you know, informal one. So thank you so much for joining us today and staying today. We'll meet again 1 week from today, January 19th, Thursday, the same time, the same Zoom link. We'll have Professor Neil Dixon from University of Manchester, again, the UK, and European country, and also Dr. Irfan Dai from Duke University. As usual, the follow-up informal chat will occur without recording. Please stay here. If you are interested in chatting with today's speakers, I will promote you to panelists who can speak and show your handsome and pretty face if you wish. And thanks. I will stop recording. Just give me 1 second. Stop recording.